Good morning to all of you. Today we are going to start the second chapter, that is structure of chromosomes. Okay. So before that, chromosomes, these are the different structures which are present inside the nucleus of any plant or animal. In the previous chapter we have seen the chromosomes. They are existing as chromatin fibers, and at the time of cell division they are coiling among themselves and they are just making the structure which we call as chromosomes. So here four diagrams are there. You can see during the interphase they are existing only as thread-like structures. You can see, and very fine thread-like structures. Here in the second phase, early prophase, you can see that they have started to condense. Okay. Here in the late prophase, you can see the structures, they are still more condensing, and at metaphase stage you can see that the total chromosome-like structure has been appeared, and here there is having primary constriction of centromere. Okay. Now, based upon the chromosomes, we'll see what is inside the chromosomes. So, inside the chromosomes, there you can see fine sheath-like structures which we call as chromonimata. Chromonimata are the subunit of chromatids, and these chromatids has been developed, or it is formed from the chromatin fibers which is making the chromosomes. Okay. So if we will talk about the chromosome, so first we should know who discovered chromosome. So chromosomes was discovered by Waldeyer in the year 1888. So in the year 1888, Waldeyer told about the chromosomes. He named it chromosomes. Okay. Then in the chromosomes, he just told that they contain the DNA molecules. They contain the DNA. The DNA is the hereditary material which is getting transferred from the parents to the offsprings, and the DNA, they are having some units which is containing some functional segments, which is the functional part of the DNA, that is known as genes. And genes are totally responsible for the characters which have been built in any of the organism, and the number of chromosomes for each and every individual, or you can say each and every species, is fixed. There will be no change in the chromosome number in any of the generations. So if it will be like so, means the species will be disturbed, or it may happen that it can destroy, and it is against the rule of nature. Okay. Now if we will talk about the chromosomes only you can see in the metaphase state, they are distinctly visible, or at other times if you will see, it will not be distinctly visible. Okay. Now see here the chromosome, how it is being packed and coiled. Here we have to study. So the coiling of chromosomes, if we will talk, we have to study the nucleosome model, where the DNA coiling, that is being studied. So the DNA, if we will talk, this consists of different parts. If we will talk about chromosomes, you can see the chromosomes, they are containing approximately 40% DNA, 50% of the histone proteins. Non-histone protein will be approximately, or it will be 8.5%, and the metallic ions will be in traces, just like magnesium, calcium, and some more will be present, might be present. Okay. Now we will see how the chromosomes or the DNAs, they are coiled. So we are having distinct, suppose this is the chromosomes, the DNA are coiled around histone proteins. These are the proteins, which gives stability to the shape of the chromosome. Histone proteins, and at the same time the histones are also responsible for the different type of enzymes, for preparation of different types of enzymes. Suppose this is a histone, and histone occurs always in octamer. Octamer you might be knowing, octa means 8 and mer means repeating unit. So 8 repeating units combine to form the octamer, and in octamer, in the shape of octamer, you can see the DNA will be arranged like this. Okay. So DNA will be arranged like this. Okay. Histone octamers. So you can tell the DNA along with histone octamer, that gives a structure that is called nucleosome. So that's why it is also called nucleosome model. The chromatin, that is it is actually a long DNA strand. It is coiled several times. The coiling is strictly according to a plan, and the plan is that in a octamer means this histone protein which is occurring in octamer shape, that is the DNA is coiled around that, and which is giving stability to the DNA. Okay, it is DNA strand is coiled repeatedly till it becomes the chromosome, familiar to us. The coiling of DNA strand can be compared to that of the telephone cord. So the telephone cord you can see, and one figure is also given. You can see the figure and you can just understand how it will be coiled. So like this is your nucleosome, or it is your histone, and this will be your DNA. So it is coiled according to this. And if we will talk about centromere, that is the primary constriction. So two chromatids will be present. This is centromere, that is also called primary constriction. So in some of the cases there will be secondary constriction also. The secondary constriction, that is generally present here. Okay, the secondary constriction is present, that is also called satellite chromosome. And the last end of the chromosome is known as telomere. Telomere is the last end of the chromosome. Okay, so based upon the structure of the chromosome, it is divided into 4 types. So based directly, it is the basis is the telomere, where the telomere is present. Okay, or you can tell where the centromere is present. So if we talk about metacentric, submetacentric, telocentric, heterocentric. Telo means at the end. So metacentric, you can see the centromere will be at the middle. So it will have distinctly 2 chromatids. If it is submetacentric, it will one will be small and one will be large. It will be just above the center position. Telocentric if we will talk, okay, so it will be having only the centromere will be at the top, so that it will have only 1 chromatid. And heterocentric, one will be very small and one will be very large. One chromatid will be very small and one chromatid will be very large. So in the metacentric, meta means middle, so in the middle it will be there and both chromatid will be of the same size. Submetacentric means it will be just above the center. So if it will be just above the center, one will be small and one will be large. And if it is a necrocentric, it will be just present, the centromere will be just present at the end, means towards the end. So one will be very small and one will be very large. And last is your telocentric. Telo means end. So that is telocentric. Only one centromere will be present. It simply means only one chromatid you can see. So this is a brief introduction about this structure of chromosome. In the next video, I will be telling you about the structure of DNA.